Anyway, now that we're recording, I will start the attendance again. So, Brian is here, Curry is not here, Garvey is here, Fisher, I believe, is online. Yeah. Hamilton, not here, is online. Hellman is here, yep. He's here. Ashley's here. One half of the Taylors are here. Crystal's here it is. And I said Taylor's, Miller's. Crystal Miller is here. Taylor Miller is online. Doris is here. Rollins, not here. Not online. Saunders, not here. Online. She's online. Shamble is not here or online. Vargas is not here or online. And Young, well, anyway. Did I miss anybody for attendance? All right, so in addition to midterms, we're going to turn in midterms this week. I also was reminded I have to do a survey for Achieve. And what you guys should know about both of them is none of them are set in stone. It's only for your information, right? So it's not like, if you ever get a scholarship or anything like that, that never shows up. So for the midterms, that's literally just so you can see what your grade is. The only people that affects are people who aren't showing up. Like literally, well, not this class, but I've had some classes where that person's never once shown up, so that's a financial aid issue. So that's another thing on their end, not your end. But for the Achieve thing, I was just trying to explain it earlier. It's hard to see. I can show you guys if you want. It's just a survey. It's got everybody's name and a checkbox. So attendance concern, if you have anything lower than an 80%, I just checked it. In danger of failing, if you have anything C or a D or below, right? Anything lower than a C, I just checked it. Same thing with low quiz/test scores. If you have 60%, D or below average for your exams, I checked it. Missing late assignments, technically, if you don't have 70 points of attendance work, I checked it, right? Because you need at least 70 points of attendance work. Technically, you were having a missing assignment if you're not up to 70 points. And then I also went through and tried to write down any labyrinths. Because I can make comments. Yeah, that's basically it. So that's, again, mostly for you. It's just letting you know where you stand. I do that anyway. You guys should know where you stand. You have access to your grades, and I like to know how you're doing. But that makes it official. You can definitely see where it is. And for some of you, it looks really bad. It's temporary, and that way somebody from the university can contact you and say, hey, I noticed you have any attendance concerns. I notice you're having low test scores or whatever. And then they can ask you, like, hey, is there anybody that can help you? I personally think if you need help, you should get it from me because no one else knows how I test, right? So, but if for some reason you want outside help, definitely get it. And that's what the university will do. That's what Achieve is for. But again, nothing to worry about. Nothing's in writing. See what's in writing. Nothing is permanent. This is going on any kind of permanent record. No one else can see it except you. It's people who work there who try to find help for people. Any questions about that? All right. Speaking of which, too, like I said, it also gives you the option to make comments. And for those of you who are like missing assignments and things like that, I left comments. So if I left a comment and said, hey, you're missing lab 6, turn in lab 6, and that means I made a mistake. It doesn't matter on that, but let me know so I can fix it. Fix your grade. So now, are there any questions about the Achieve thing? And again, you'll get an automated email from that. If you need help, please get it. All right. Still recording. Let's jump into the rest of this chapter. First word for attendance. If we, let's see. Let's just say parents. Or parents, either way. Plural, so you can know if you're a parent. So again, if you're online, you have to send these words. Even if I said, yes, you're online when I took attendance, you still need to send these words before 9 a.m. Anyway, wait, this is way past what we left off. We already talked about the dihybrid cross, we already talked about the test cross, we already talked about pedigree, talked about recessive disorders, talked about dominant disorders. Skip this part, process of science, you can read it, it's good stuff. We briefly flew through this because there's not going to be questions about this on the exam. There are questions about this on the study guide. There we go. That's where we left off. Now here's my favorite part of the chapter, because this is the most realistic part of the chapter. So think about all the stuff we did in lab, where it was like one gene controlled one trait, or excuse me, one character, and that one character usually only had two traits. So for example, there's one gene that says you either have a smooth chin or dimple chin. Really simple, right? But there's one gene that said you either have attached earlobes or unattached earlobes. The thing is, all the stuff I've taught you is not very common. Usually, there's more genes that affect one thing. Maybe one thing that affects multiple genes. Maybe there's one gene, and instead of having two phenotypes, like smooth or dimple chin, there's a number of phenotypes. Like think about eyes. Even though we did eyes in lab, think about it realistically. It's not just blue eyes or brown eyes, right? There's blue eyes, brown eyes, different shades of blue, different shades of green, so on and so forth. So this is what we're getting into now. Or as the book says, Mendel's laws that we just learned about in the past two readings. They explain inheritance in the terms of genes passed from generation to generation according to the rules of probability. So the rules of probability, all that stuff still stands, right? This is still valid for all sexually reproducing organisms, which is what we are. But, this is what I was getting at right here, it does not explain some patterns of inheritance. And that's what we're going to end the chapter with, is talk about these other patterns of inheritance. And like I previously said, mostly Mendel's laws can't account for these patterns that we're about to talk about. But let's get into it. And again, this is in a slightly different order than your book teaches it, and I'm going to use different examples in some cases than your book teaches it, but that's okay. Remember, I'm never going to ask you questions about the examples. You need to understand the concept. Speaking of which, I'm about to introduce you to different types of dominance. So far, you should know by now the difference between recessive and dominant, right? So again, if you have one dominant allele for cleft chin, then you're going to have a cleft chin, right? It doesn't matter if you have one allele or two alleles. That's what makes it dominant. What I haven't told you is that is actually called complete dominance. So all the stuff we've been talking about so far is called complete dominance. And now I'm about to give you some other examples of stuff like incomplete dominance. And even though your book is it's not put this way, there's something called codominance that we're going to talk about. Pleiotropy, polygenetic, it should be polygenic inheritance, epigenetics. So there's all these different variations to what you learn. And most of the questions on the exam are going to be me making up some hypothetical mating, and then me telling you what the offspring are. And from that, you need to determine whether it was complete dominance, which is what I've already taught you, or this other stuff. So, for example, if I said, a blue alien and a yellow alien mate, and 100% of the offspring are blue, then you might say, oh, that's complete dominance. Blue must be dominant to yellow. Again, think about that when we move forward and talk about these other examples. Incomplete dominance. This is when the F1 phenotype is between the two parents. So you need to know that. Not the definition of that, because I'm not going to ask you the definition of incomplete dominance. Again, I'm going to give you a hypothetical mating, and then you need to recognize the outcome of that mating and say, oh yeah, that's incomplete dominance. So for example, if I say a white flower and a red flower breed, don't read that yet. If I say a white flower and a red flower breed, and 100% of the offspring are red, and that's complete dominance, right? That's the stuff I've already taught you. If I say a white flower and a red flower bred, and we have a pink flower, then you should say, oh yeah, that's incomplete dominance, because it's merging the two phenotypes. Does that make sense? With complete dominance, is one phenotype or the other. One is dominant to the other. With incomplete dominance, they blend. So a black mouse and a white mouse gives you a gray mouse. Something with curly hair, something with straight hair gives you wavy hair. So it's always an intermediate. If it worked like this for humans, and no, I don't want to use that example. Again, And I can just make up something and say a blue alien and a yellow alien mix and mate, and all their offspring are green, right? Because when you mix blue and yellow with green, that would be incomplete dominance. Does that make sense to everybody? All right. Here's some pictures from your old textbook. As the example again, red flower and white flower. If this was complete dominance, and you can see here, for some reason, they just chosen red as dominant. If that was complete dominance, then all those offspring would be red. But it's not, right? It's incomplete dominance. So they both contribute. And not that you need to know this, but if you're curious as to why that is, this isn't 100% accurate, but it's accurate a lot of times. It's simple enough for you to understand it. In the old situation, where you had a red flower and a white flower, and they were all red, that's usually because, excuse me, the allele, this allele, the big R allele, said, hey, make red, right? And in this case, the small R allele, the small allele, the recessive one, was just a broken gene that didn't say anything. So therefore, if you have a gene that says make red and a gene that doesn't say anything, and you mix the two, obviously red's going to show up. But in this case, this allele, we're talking about this actual case, this allele, it's not a broken gene. That one actually says make white. So here we're mixing red and we're mixing white. That's what makes it pink. Not that you need to know that much detail, but hopefully if you're like me, that's just you're wondering, well, why? How does this happen? But anyway, those are the outcomes. And then your old textbook goes on even further, because this is like the situation, right? We always talk about P generation, and then we talk about the F1 generation, and we talk about what happens if we cross the F1 generation. And here we go. We still get the same genotype ratio. We still get one homozygous dominant. We still get two heterozygotes. And we still get one homozygous recessive. Except now, because we have incomplete dominance, we don't have three reds and one white. We have one red, two pinks, one white. But again, for the exam, I'll focus on everything above that line. I'm not even going to get into the F2 generation if I'm trying to have you distinguish between complete dominance or incomplete dominance or the other ones I'm going to teach you about. So any questions about that? All right. Here's a book example from your old textbook. You don't need to know about it. It's just an example, but it's a real life example and it involves humans because really in the grand scheme of things, flowers aren't that important. So, well, they are. But anyway, hypercholesteremia, hypercholesterolemia. That's basically when your blood pressure or blood cholesterol is too high. It's a good human example of incomplete dominance. When you have it, if you're heterozygote, so not even homozygous dominant, if you're heterozygote, your cholesterol level is 2 times more. For homozygotes, it's usually about 5 times more. And a lot of times these people have heart attacks as early as the age of 2. So again, this is just an example. So if you're studying this, you don't need to memorize any of that. But let's talk about how that happens. So in this case, it's because this kind of goes opposite of what I said previously. But in this case, the dominant gene says, hey, you need to make these things that pull cholesterol out of your bloodstream. And the recessive allele, is just a broken gene, so it doesn't say anything. So in this case, right, if you're homozygous dominant, that means you have the ability to have these things that take the cholesterol out of your blood. If you're homozygous recessive, that means you're just not making any, right? You're not making any of those proteins that pull the cholesterol out of your blood, so you have way too much of it. If you're heterozygous, again, you still have at least one allele that's making these proteins, so you are pulling some of the cholesterol out of you. And again, this is just an example. You don't need to know it for the exam. There might be questions about it on the study guide. I don't remember. I should take another look. Anyway, the next word for attendance is mild. Any questions about incomplete dominant? I'm not going to make it tricky on the exam. What you just need to remember is there's a mating. The offspring are a blend of the two. That's incomplete dominance. All right, next one we're going to talk about is codominance. Your old textbook goes straight into the example. So I'm going to talk about the example quickly, but I'll go ahead and tell you, I'm not going to ask you any questions about ABO blood groups. I have questions about it on the study guide, but for the exam, I'm not going to ask you about the ABO blood groups. How many of you guys know your blood type? So some of you, right? I do too. And that's what this is, right? So like I'm O positive. So if you know your blood type, you're going to learn the basics of what this means. This is interesting because not only is it codominance, which is what you're going to be tested on, codominance, not blood groups. Another thing that makes this interesting, and this is why I stuck with it, is also now we're dealing with 3 alleles. So far, all the alleles we've been dealing with have been 2. It's either dominant or recessive. So in this case, even though I'm only going to teach it now, and then we're going to be done talking about 3 alleles. But in this case, we have 3 alleles controlling, or 3 alleles of a single gene. So these 3 alleles produce these 4 different phenotypes. In this case, the phenotypes are blood types. So you're A, B, AB, or O. And these letters refer to carbohydrates. So this is going to make more sense than I should have. On your blood cells, on the outside of your blood cells, you have carbohydrates. This way, your body can help identify it. Like, does this blood belong in my body? Let's talk about it really quickly. For example, if you are blood type A, that means you have carbohydrate A on the outside of your cells. Therefore, your blood is anti-B. Your blood, your body says, hey, only A belongs in here. Anything with a B in it, we know it doesn't belong, and therefore it's not good, right? So it can take A, obviously. It can take O. I'll talk about that when we get there. But obviously, you can't take B because B has the B carbohydrate on it. Then it can't even take AB because even though it has A, it still has B. The body says, nope, can't do that. Carbohydrate B is the opposite, right? Carbohydrate B is anti-A, so it can't take any kind of blood that has A on it. AB is what we call the universal acceptor, right? Because it has carbohydrate A and carbohydrate B, so it can take it all, right? It doesn't matter. It's not anti-anything. And then the opposite of that would be blood type O, which is what I am. That's the universal donor. Because there are no carbohydrates, right? So your blood type A, you're not anti-O because there are no carbohydrates at all. Same thing here. If you're B, not anti-O because there are no carbohydrates on it. So again, AB is the universal acceptor because it's not anti-anything, and O is the universal donor because it doesn't have any carbohydrates. And then on the flip side, because of that, O is like anti-everything other than O. So if you have O blood type like I do, it's really helpful for other people, but it could put you in a pinch, put you in a jam, right? Because I can only have O blood types. So really quickly too, let's talk about this. Like I said, there's 3 different alleles. Actually, no, I'm going to skip that. Because I don't want to confuse things. I'm not going to use 3 alleles in the exam. So we won't even talk about the genotypes. I'm going to give you some other examples of codominance. But there's that. Any questions about the blood groups? For independent work, if you want to, you can get into this since I'm skipping it. Right? So you can see O is homozygous recessive. And so on and so forth. It gets different, right? Because, again, there's 3 alleles. There's A, which is dominant, there's B, which is dominant, or there's little I, which is obviously recessive. Another thing you can look into for independent work, because I haven't mentioned it at all, is pluses or negatives, right? So I am O positive. Some of you guys might be A negative or whatever. So we didn't even talk about the plus and negative. But look that up if you want to for independent work. This slide is everything I just said, but in writing. Same with this, right? It's everything I just said, but in writing. Same with this, everything I said, but in writing. It brings us to this very important word. This is what I'm getting at. This is what you need to know for the exam. Is we're talking about codominance. So here's some examples like you're going to get in the exam. So again, let's back it up. The first thing I taught you about was complete dominance, right? And in that case, one is completely dominant to the other. So if you mix a red flower and a white flower, for example, get all red flowers, that's complete dominance. And then there's one we just talked about prior to this, which was incomplete dominance, right? If you mix a red flower and a white flower, they blend, right? And when you mix red and white, you get pink. So that would be incomplete dominance. Codominance, and this is usually where people screw it up on the exam. I can see why it can be tricky. Codominance is both phenotypes individually show up. So they don't blend, right? It's not like you're mixing red paint and white paint and you get pink paint. They don't blend. Both phenotypes show up. And that would be like this picture here. A red flower with white spots, or a white flower with red spots, however you want to look at it. So again, I'm going to put it next to all this. These are just examples. I'm not going to ask you a specific example about these cows and those flowers. And actually the dog thing is not even a real example. I just made that up because it's easier to visualize. A white dog and a black dog gives you a gray dog. Or, excuse me, gives you a black dog with white stripes, white spots, or a white dog with black spots. So those are just examples. The point is, in the exam, I might give you an example, or let's say something like the yellow alien mated with the blue alien, and all the offsprings were yellow with blue spots, right? That would be codominance. Meanwhile, if I said all the offspring were green, that would be incomplete dominance because they blended. If I say all the offspring were blue, that would be complete dominance. Blue would be completely dominant. So any questions about codominance? All right, so those are the big ones that are going to be like, on the exam where I'm like, all right, these things made it. Here was the results. Then you have to choose. Was it codominance, incomplete dominance, or... All right, let's talk about something else, a new thing here, something called pleiotropy. So this is new too. I mean, new to our discussion. Everything I've told you so far has been one gene controlling one character. Even when we're talking about blue blood type, yes, there was 3 different alleles, but it was still one character, right? The character was blood type. And there was 4 different phenotypes, 4 different traits. Here, we have one gene and several characters. And a great example of this is sickle cell disease. So one gene causes sickle cell disease. We're going to talk about that gene later too. We're talking about mutations, we're talking about evolution. This is going to come back. But anyway, this is a good example. That might be on the exam. In this case, when I'm asking you about pleiotropy, might just ask you to remember the exam. No, I take that back. Now, I'll give you another example. So in the exam, if I say something like, there's this child inherited this gene, and now because of that, and I have a whole list of things. If there's a whole list of things based on one gene, right? That means pleiotropy. Anyway, really quickly, talk about sickle cell disease. It results in abnormal hemoglobin proteins, which causes your red blood cells to deform into this shape right here. So instead of looking like a round donut like it should, it kind of looks like it has that sickle shape. I'm going to put it next to this because, again, I'm not going to ask you questions about sickle cell disease. It's just a great example. Here's what it looks like, or kind of a flow chart. So if somebody is homozygous for the sickle cell disease, that means that the first trait, the first character that's messed up, is that you have an abnormal hemoglobin, right? That's the first character. But due to that, you also have an abnormal blood shape. Due to that, you have all these other symptoms that go along with that disease. So again, for the exam, to understand what, to know what pleiotropy is, it's one gene and a lot of characters. Everybody remember that? One gene, a lot of characters, pleiotropy. That in itself is not too hard, I don't think. And remember, it's multiple choice. So if I were sitting in your seat right now, then pleiotropy, no, I don't remember that. But I don't have to remember that. You just need to recognize it when you see it on the multiple choice exam. But here's where it gets harder. The next one sounds similar, yet it's the opposite. The next one we're going to talk about is polygenic inheritance. That's misspelled. There we go. Polygenic inheritance is the opposite. And your textbook calls it epistasis. Know them both ways. Your exam might use either word, either one. Maybe I'll put them both, I don't know. But instead of having one gene and many characters, this is the opposite. This is 2 or more genes affecting one character. So for example, again, if we were to do a bar graph of what we did in lab, and it was like, all right, the bar graph was for chin dimples. Like that many people had it, this many people didn't. Or if it was for blue eyes, right? This many people didn't have it, this many people did, right? If you were to do a bar graph for what we did in lab, there would only be 2 bars. Does that make sense? You either have the hitchhiker's thumb or you don't. You have the bent finger or you don't. You either have hairy middle knuckles or you don't. You guys with me so far? Does that make sense? Because that's the character. It's controlled by one gene. So that one gene has two different phenotypes. But now what we're dealing with is a lot of genes. Therefore, when you graph it, what it's going to look like is this. A bell curve. And this is really oversimplified, but height, this is an example. The example here is height. And the height is a very good example. Most people on Earth, I think, are 5'6". I don't know for sure, but that's where it would be, right? So if you were to graph our class, we wouldn't have 2 bars. How many people were here the last time? 13? Right? I think there was 13. There would be probably 13 bars, right? Each person would have their own bar because height varies that much. It's not like humans are just 6'6" or 5'6", right? Or it's not even like they're 6'6", 5'6", or 6. It's a whole wide range of heights, from really short to really tall and everywhere in between. The reason I'm focusing in on this bell curve thing is because that's how you can determine if a character is controlled by 2 or more genes. When you graph the phenotypes, we get a bell curve. Any questions about that? I know it seems complicated, especially when you look at this huge Punnett square. And it is complicated if you were to actually study it. But we're not actually studying it. I'm just telling you, polygenic inheritance. More than one gene controlling one character. And the way you can recognize it, again, if you were to graph it on a bar graph, you get a bell curve. And again, I want to point out, even this is very simplified. You can imagine this could really be broken up into every little tiny height. Here's someone who's 6 foot. Here's someone who's 6 foot and 1/2 inches. Here's someone who's 6 foot 1 inch. So far, so on and so forth. And another thing, height. And you can look into this if you want for example. I don't think we've nailed it down yet. I think we're still trying to figure out how many genes are actually involved, and I think it's way more than 3. Even consider this. This is only they've simplified this and said 3 genes. So when you do a Punnett square with 3 genes. So remember, we've talked about a monohybrid cross. We did a dihybrid cross. That's a trihybrid cross. Imagine 4 or 5. So anyway, great example. Another example for this would be IQ, right? IQ, again, we don't know all the genes involved in that either. It's a wide variety, and it would be a bell curve. Probably very few really dumb people, probably very few really smart people, and a lot of very average people. That's another thing you can look up for independent work if you want. What are some other good examples of polygenic? Or for that matter, anything that we talked about, any of this stuff. So see if there's other examples that I haven't given you of incomplete dominance, or complete dominance, incomplete dominance, codominance, myotropy, polygenic inheritance. But let me back up one more step too. Another thing I can tell you about height, not that you need to know this, but this is a good segue. What we do know about height is it's not 100% about your genes. It also has environmental factors. So if you were to have 2 identical twins, separate them at birth, and one of them had a really good, healthy life with good nutrition, and the other one didn't, well, the identical twins are going to be different heights. Sometimes, very often, it's not just your genes that control stuff, it's also environment. That's what we call epigenetics. So, many characters result from a combination of heredity and environment. So, in short, even though I'm about to tell you a bunch of stuff, taking notes, you should just epigenetics equals environment, or genes plus environment. So now we're not just considering just genes, we're also considering the environment. And this kind of goes back to the question that Crystal asked me earlier this week or last week or whatever it was about the mutated gene and those DNA tests you can get. So you might, I'm being really vague here because I don't know exactly which gene she was talking about, and I don't remember which gene specifically does these things. But you might have a gene that's mutated, which might make you more likely to get a certain type of cancer, but also your environment plays a role. So you might have a gene that makes it more likely, but the environment might turn a certain gene on or turn a certain gene off, in which case, that's when it's activated and not activated. Anyway, let's jump into this. And also the next word for attendance, I'm just going to circle it. I'm not going to say it this time. No. Anyway, the question of whether or not certain traits are influenced by genes or environment, or as you hear often, nature versus nurture, is a very old and hotly contested issue. This is especially true these days when it comes to things like gender. Not sex necessarily, but gender, right? There's a lot of talk about gender these days. Back in my day, the big thing was homosexuality, right? It's like, is it genetic or is it environmental? These are all things you can look up for independently too. Or anything you can think of that you might find interesting. Some of you are athletes, right? So is athleticism genetic? Is it environmental? Is it influence of growth? Musical ability? Humor? Intelligence? I don't know. Anything you can think of that you're interested in that's worth looking up and see. Is it environment? Is it nature? Or is it nature, nurture, or both? Anyway, I've already used this example if you talk about identical twins. How many of you do know identical twins? Like know them, not just know of them, like friends with them? You do? Can you tell the difference between the two of them? No? How long have you known them? Oh, wow. So that's a bad example because usually, I should say usually, sometimes you can't tell the difference because, I'm trying to explain this because this is going to be important later, not necessarily for this chapter. If you think of your genome as a book of instructions on how to build a protein, right? On how to build proteins, that's what it is. You can think of identical twins as having the same exact set of instructions, right? So if you were to look at page 85, and it tells you how to build an eye, if you were looking at his book and then look at his book, go to page 85 and look how to build an eye, you have the same exact instructions, right? But if you were looking at page 85 for me, page 85 for her, right there it would be instructions on how to build an eyeball, but mine would be slightly different than her, right? Because we're not related, we have different eyeballs. But for identical twins, they would have the same exact instructions. So the same exact copy, the same exact book. The difference here, we're talking about epigenetics, we're talking about environment, is even though we have the same book, so to speak, doesn't mean all those pages are being able to be read. You ever open up, like put a book down and open it up, it just kind of flops open to a page because maybe you put it down upside down or whatever. You know what I'm talking about? It's kind of the same thing with your genome, right? Sometimes certain genes for certain people are easier to be read because of whatever environmental factors played a role in the chromosomes. Or I'm sure you guys have seen books where the pages are stuck together, right? Since it's the same thing here, right? So in your genome, sometimes genes are harder to be read because of environmental factors. So that being said, in general, it's only the genetic influences that are inherited, and the effects of the environment are generally not inherited, generally speaking. So again, twin A, these two twins might have a disease or a gene for a disease, and maybe twin A has the disease, and twin B doesn't. Because maybe twin A, some things happened in the environment that turned on that gene. But for twin B, even though they have the instructions, they have that disease, they have that gene, it wasn't turned on. Right? But that doesn't mean that twin A is going to pass along the disease more than twin B, right? They have the same gene. That's what this book would say. However, it gets trickier again, because in recent years, biologists have begun to recognize the importance of epigenetic inheritance. So far we've just been talking about epigenetics, and for the exam, that's all you need to worry yourself about. Epigenetics, again, we're talking about genes and environment being mixed together. But for your own information, epigenetic inheritance it's a whole different thing. So now we're thinking in some instances, sometimes things that you do and the environmental factors that you go through, sometimes that can be passed down to your offspring. So for example, I don't know, that's a bad example maybe, but it's easy to imagine. It's true. If you imagine somebody smokes a lot, you're thinking, oh well, I'm smoking now, but I'm not going to smoke when my kid's born. I'm not going to smoke around them. But using that as an example, what if the smoke messed up your genome so bad that even that environmental influence was also passed down? Anyway, as your book or the old book defined epigenetic inheritance, it's the transmission of traits by mechanisms not directly involving the DNA sequence. So again, it's not the alleles, but remember when we talked about the histones, we talked about what chromosomes are made up of, it's DNA wrapped around proteins, and those little proteins, those little balls we call histones, and Those things are called nucleosomes, and those wrapped around it. And I said this will be important later. Well, here it is. This is what I'm talking about. So there's certain things about your DNA that we don't even get into in this class that make certain genes easier to read or harder to read. And that's what I'm talking about with environmental influence. And you can look it up for independent work if you want. It can get a little tricky. But look it up if you want. Anyway, here's an actual example from your textbook instead of just someone that I made up. Chromosomes can be chemically modified by adding or removing chemical groups on the DNA or the protein components. So that's kind of what I was getting at, right? The DNA is literally wrapped around these proteins. And that can make it easier or harder to read a gene, or in a sense, turn it on or off. An environment plays a role in these changes. That may explain how one identical twin can suffer from a genetic-based disease versus the other doesn't. Sorry, we talked about that earlier. I gave you that example, twin A versus twin B. So simply put, epigenetic changes are changes to the DNA and chromosome, while genetics are strictly the sequencing of the gene, right? So your A's, your T's, your C's, and your genes. All the other stuff we're talking about is actually changing your genome. Epigenetics, we're talking about changing not just the genome, not just the letters, right, but the way it's all put together. But again, for the exam, this is not very important. It might be like one question about it, but it's important for your everyday life later. Again, if you ever get a disease or have to deal with somebody you love having a disease. Epigenetic modifications and changes in the gene activity that result in those changes might be carried on to the next generation. I already alluded to that earlier when we were talking about epigenetic inheritance. But the good news is, as far as we know, unlike alterations to the DNA sequence, these epigenetic changes can be reversed, as far as we know. So, for example, if you like to sunbathe a lot, and the UV rays mutate your actual DNA and change your code, and that happens in your reproductive organs, that cannot be reversed, right? That will get passed down when you produce your sperm or your egg. But if you just made modifications to your epigenetics, right? Modifications to the proteins, chemical groups that are added to your DNA, that can be reversed. And again, I'm not going to test you on any of this stuff, but it's really, really worth looking into for independent work. So your next word for attendance, I'm going to underline, and I'm not going to say it. Next word for the next portion of the chapter is going to go very quickly, which is good because I'd like to finish the chapter today. It's basically, I'm going to repeat everything we've already said, except now we're going to look at it from the point of view of chromosomes, because that's how your old textbook did it. However, when I was having these discussions earlier, I also was having a discussion about chromosomes, so drawing them on the board. So again, everything I'm about to tell you is what I've already told you, but from the point of view of chromosomes. The first thing we're talking about is linked genes. The chromosome theory of inheritance states that genes are located in specific positions on chromosomes. You already know that. Remember, I showed you a picture. There's like two different There was two different chromosomes on the side. One from your mom, one from your dad. They were kind of like sideways like that. Obviously, they were different colors because one was from your mom, one was from your dad. And then had a little thing like, all right, there's gene A. There's gene, I don't remember for sure, I don't know, gene B. Here's gene C, whatever it was. And it had little arrows and said that's the gene loci. So we've already had this discussion. But yeah, genes are located in specific positions or loci or locus or singular on chromosomes. So again, as a reminder, if the gene for stinky feet, I'm making this up, if the gene for stinky feet is on chromosome number 17 at the very tip in my genome, that means the gene for stinky feet is at the same place on all your genomes, right? Chromosome number 17 is at the very tip. That's how it works. And as I've already said in the last chapter, and I've been bringing it up in this chapter too, behavior of the chromosomes during meiosis and during fertilizations accounts for these inherited patterns. So again, everything I just finished teaching you for this whole entire chapter all boils down to what the chromosomes do. Because it's the chromosomes that undergo the segregation, right? So they separate, right? And they also have that independent assortment. So again, to remind you, independent assortment is like saying, It's not like all your mom chromosomes line up on one side and your dad chromosomes line up on the other, and those are the things that separate during meiosis, right? It doesn't go like that. Right? So when you make sperm and you make eggs, it's not like half of your sperm have your mom chromosomes, half of your sperm have your dad chromosomes, right? They mix up. That's independent disorder. Here's a visualization of what we're saying. And again, we're going back to the P. Here we're looking at two traits, P color and P shape. This one happens to be smooth and yellow. This one happens to be wrinkly and green. This is just an example. You don't need to memorize this for the exam. But again, you can see, we've already talked about this. I'll come back to let's go through this. This probably confuses people. You've already seen this, right? So this goes back, here's the independent assortment I was talking about. When this thing goes to meiosis, yes, it could be all mom chromosomes on one side, all dad chromosomes on the other, or it could be the other. And keep in mind, here, this is really oversimplified. We're only looking at two homologous chromosomes. Meanwhile, humans have, two pairs of homologous chromosomes. Meanwhile, humans have, 23 pairs of homologous chromosomes. So imagine this, but with 23 pairs instead of just two. But anyway, even with just two, you can see with all mom on one side, all dad on the other versus both versions of that. When those things go through meiosis, you have all these different varieties, all these different possibilities. There's a gamete with all mom. There's another gamete with all mom. A gamete with all dad. Another gamete with all dad. Gamete with both. Another gamete with both. A gamete with swap both. So on and so forth. Again, your book is just trying to reiterate this idea of independent assortment and why it's so important to give us variety, which is important for sexual selection. And then, same picture, a little bit more information. That out of the way, again, everything we've talked about is about chromosomes. Let's talk about some new information. Linked genes. Linked genes are located near each other on the same chromosome. So that's going to be a test question, probably. It's definitely a question on the study guide, I think. What are linked genes? You can build the definition. They are genes that are located near each other on the same chromosome. So, let's talk about, and this is all about crossing over. Draw the other chromosome. So imagine this is what you're looking at in prophase 1, right? So we're talking about the first prophase, right? The first meiosis. Homologous chromosomes are together. Let's say you got this one from your mom. Let's say you got this one from your dad. There's gene 1. Here we go. Gene 2 and 3, they're kind of close together, right? You guys can see that. Oh, right? You can see how close genes 2 and gene 3 are together, right? But now let's imagine there's crossing over, and we swap a chunk. So in this case, let's say the crossing over happened right here. That's where the crossing over happened. So this chunk swapped with this chunk, and this chunk swapped with that chunk. In that case, gene 4 is going to be changed, right? So try to picture in your mind the dad version of gene 4, dad allele, is now going to be on the mom chromosome. And mom's version of gene 4 is now going to be on the dad chromosome. You guys picture that, crossing over? And that's what happens. I told you that's what crossing over is. That's nothing new. But when genes are really close together, then they do go together, right? So when these things go their separate ways in meiosis, if you have the gene 2 from your mom, you're most likely going to have the gene 3 from your mom. So again, let's just say, talking about hairy armpits, hairy knuckles. If gene 2 is for hairy armpits and gene 3 is for hairy knuckles, and your mom had hairy armpits and hairy knuckles, then most likely, if you get her hairy armpit gene, you're also going to get her hairy knuckle gene. Because they're right there next to each other. So if you get those separated, right, and you get that chromosome, you're probably going to get those two genes because they're close together. Yes, sometimes they're swapping. Sometimes they swap. You have crossing over. That might be different, but gene 4 is far enough away where it probably won't affect. And you're probably like, I don't care. What does this matter? I give you the information you need to know. If you see something on the exam other than a 9 to 3 to 3 to 1 ratio, then that should automatically, you should think, oh, those must be linked genes. If you see something different than 9 to 3 to 3 to 1, it's linked genes. This is the thing that has a whole chapter. Well, first of all, when you're a biology major, don't do one chapter called genetics. We have a whole class on genetics. And in that whole class on genetics, this whole idea about linked genes has a whole chapter. So it is actually really complicated, really interesting, slightly important, but we don't have the time or the expertise to really dive into it. So again, Cliff Notes versions, if you were to see some ratio, some 4-digit ratio like this that wasn't 9331, you should think of those as linked genes. And definitely do the study guides. The study guides has questions on this. So do those questions. And if they don't make sense to you, then ask me. Because I think that's the best way to teach you, is to actually look at a problem and me to explain how to fix it, how it makes sense. Anyway, any questions about linked genes? All right, the next one we've already talked about, so we're going to do this very quickly. Sex determination. Long story short, we've already said this. Last time I said it was for all mammals. But obviously, since humans are mammals, the same is true. What you need to know, males are XY, females are XX. That's it. I've already talked about this before. Are there any questions about that? Here's what they look like. There's the X chromosome. There's the Y chromosome. Therefore, we're looking at a male right there. That's a male's genome, male sex chromosomes. Which brings us to this, sex-linked genes. As you might imagine, the sex-linked gene is a gene that is located on the sex chromosome. You need to know that for the exam too. Most sex-linked genes are found on the X chromosome, not the Y chromosome. Any guesses why? Don't overthink it. Use some common sense and look at that picture. Why are most sex-linked genes found on this chromosome and not that one? And well, let's just go back to my analogy and think of chromosomes as a book of instructions on how to build a protein. Here we have a really big book. Here we have a tiny book. Right? So it just makes sense, even without you guys having a lot of knowledge about this, just look at it. It's a lot bigger. So there's going to be a lot more genes on this than there are on that. That probably won't be a test question. This is for your own knowledge. Some human conditions, including red-green color blindness, hemophilia, and a type of muscular dystrophy, result from sex-linked recessive alleles. You don't need to know that. I'm not going to ask you which conditions are sex-linked. It's good to know for other reasons. This is not a pathology class. This is not an anatomy and physiology class. So you don't need to know those, but those are good examples. Matter of fact, since we're running short on time, I'm not even going to explain it. Red-green color blindness. I'm not going to explain that. I'm not going to have any questions. How many of you cannot read that number? So everybody can read the number. You can't read it? So it might be just due to the poor lighting, or you might have red-green color blindness. This is a 7. Can you see none of that pointed out? Okay, that's probably just it's not you then. It's not you, it's me. Well, anyway, if you were to get an actual test and you couldn't see that, then you probably have red-green color blindness. But the important part here, at least for this example, is because they're located on sex chromosomes, sex-linked genes exhibit unusual inherited patterns. I don't want to rush through this, so what we'll do is on Monday, we'll talk in depth about how sex-linked genes, the inherited patterns of those, and then we'll jump into the next chapter. So there we go. You guys have 2 extra minutes for your weekend. And weekend will be the last word for attendance. Weekend. That's it. Excuse me.